Good afternoon, everybody. Thank you for joining us today for Metabolomics and Life Sciences, where you'll get a chance to learn about the field of metabolomics and how Alberta has become a world leader in metabolomics from Dr. David Wishart from Canada's National Metabolomics Facility based right here in Alberta. My name is Trish Filovich, and I'd like to take a few moments to talk about BioAlberta. BioAlberta is a member-driven and funded not-for-profit industry association representing roughly 200 members of Alberta's life sciences industry. We were founded in 1998, and BioAlberta is the central voice and organizing hub for the life sciences industry in Alberta, actively driving the vision of a thriving and global competitive life sciences industry that fuels sectors of Alberta's economy. Our activities are focused on advocacy, promotion, and proactively facilitating the growth of Alberta's life sciences industry. We also serve as a voice and connector for member organizations and the broader innovation ecosystem. Our mission is to promote and grow the life sciences industry in Alberta by creating a network of companies that can support each other. We help companies by understanding their needs and challenges and navigating them to other members of the life sciences ecosystem who offer services or support. These connections provide members with business support and expertise, be it guidance from a policy, regulatory perspective, understanding the Alberta's life sciences ecosystem, access to capital or investors, and much more. It is my pleasure to introduce to you today Dr. David Wishart, co-director of the Metabolomic Innovation Center. Dr. Wishart is a Yale graduate and he's a distinguished university professor in the departments of biological sciences and computing science at the University of Alberta. He also holds adjunct appointments with the Faculty of Pharmaceutical Sciences and with the Department of Pathology and Laboratory Medicine. He's been with the University of Alberta since 1995. His research interests span many areas, including structural biology, bioinformatics, prion biology, nanobiology, and metabolomics. From 2006 to 2009, Dr. Wishart led the Human Metabolome Project, a multi-university, multi-investigator project that cataloged all of the known metabolites in human tissues and biofuels, fluids, using advanced methods in NMR spectroscopy, mass spectrometry, multidimensional chromatography, and machine learning. Dr. Wishart and his colleagues identified or found evidence for more than 8,500 endogenous metabolites. This information has been archived on a freely accessible web resource called the Human Metabolome Database. And on that note, I'm going to turn it over and have Dr. Wishart begin his presentation. Thank you, Dr. Wishart.
Thanks very much, Trish, and welcome everyone for this afternoon. I'm not sure if we still have to admit more people into the group or not, but I'm going to share my screen now, and hopefully everyone can see that first slide. Trish, can you confirm that it's A-okay?
Yep, I'm just going into the view options here. Hang on.
Okay, so as advertised, this is the first of 4 presentations that we're doing on metabolomics and life sciences. It's a series. This is, I guess, the anchor session. We'll have 3 others in the areas of precision medicine, environment, and agri-food. But this is to give people a bit of an introduction and to also tell how metabolomics can be used to move some of your discoveries or your ideas towards translation and potentially some business opportunities. I'm going to at least give a bit of an introduction to the different ohms of life. I think most people are familiar with the genome and genomics, and that usually takes, I guess, most of the air in the room when people talk about advances in omics technologies. So the genome, of course, is the DNA that you'll find in all of our cells and bodies. Genes are DNA code for proteins. The collection of all the proteins in a cell or an organism is called the proteome. And proteins actually code or help carry out enzymatic transformations, and they're responsible for producing the small molecule metabolites. And the collection of those metabolites is called the metabolome. So it's genes codes for proteins, which then codes for metabolites. And it's the genome, proteome, and metabolome. When we talk about the metabolome, we often think about collections of metabolites or endogenous molecules, things like carbohydrates or amino acids or organic acids. But as we've explored more and more, especially looking at the human metabolome, we start to understand that there are other things. These are exogenous or xenobiotic compounds. These could be food additives that give various food products their color or even their flavor. They could be things that you breathe or drink that are in the form of pollutants or surfactants. They could be drugs or supplements that you may take in the morning or that are prescribed to you. They could also include cosmetics, from perfumes to dyes to the things found on your clothes. All of those chemicals actually end up in your body. They also end up in the water, and so they end up in your dog or your cat and many other animals and plants. So the metabolome is actually a pretty diverse collection of compounds, and it seems to have a pretty important role in not only human health, but animal and plant health and well-being, as well as our environment. One way that we tend to look at metabolomics and other omics is this pyramid, with the base of the pyramid being the genome. And as I highlighted before, the genes code for the proteome, and the proteome codes for the metabolome. And it's not as if the number of molecules decreases as you go up, but it's just simply, that's the way a pyramid is built. Obviously, the methods to study the genome, we call it genomics. The methods to study the metabolome is metabolomics. One of the reasons why we draw this pyramid is that a single base change at the genome level can have a profound effect at the metabolome level. I don't know, Joy, I think your microphone is on, if you wouldn't mind turning it off. Anyways, the influence of the genome on the metabolome is also mirrored in differences or influence of both environment and physiology. So at an environmental level, the genome is almost unchangeable. What you eat or drink is not going to affect your genes. Hopefully it doesn't. What does influence metabolome is the environment and what you do eat or drink. And that can lead to significant effects on the level or number of metabolites we read. Same thing that we see in terms of physiological influence. The genes in your liver or your brain or on your skin are all identical, but the metabolites found in your liver or in your skin are all quite different. And so as you go up this pyramid, so is there an influence in terms of physiological effects. So the fact that the metabolome lies at that interface between the genome and the environment makes it uniquely sensitive. And that's why I put a little bird at the top, to really indicate that the metabolites, the metabolome, is the canary of not only the genome, like the canary in the coal mine, but also the canary for your environment. And so that makes metabolomics really useful. And this is what people are starting to realize now. The other thing that influences the metabolome is the temporal effect. Again, if we were to eat a big meal, some of you may have just finished lunch, hopefully what you ate is not mutating your genome. So over time, what you eat, breathe, or drink does not affect your genome. If you had a big load of carbohydrates for lunch, it would have changed a few proteins, probably glucagon, insulin, maybe a couple of other small peptides. But by eating a big plate of spaghetti, your metabolome is changing and continues to change. It changed within seconds of your first bite, and it will continue to change up to hours after while you're digesting your meal. In some regards, people might view that very significant time response for metabolomics as problematic. It can be in certain cases. On other cases, the rapid changes to your metabolome are absolutely essential for your body to adapt, and being able to measure those short timescale events are actually pretty important for understanding biology, physiology, and health. So there are differences not only in what we see and measure, but also in the timescales between genomics, proteomics, and metabolomics. Now, when it comes to measuring the metabolome, there's sort of a standard protocol or pathway. We often begin with samples, biological samples. That could be plants, it could be cells or microbes, it could be tissues, organs. Usually those samples are solid. So what we have to do is we extract them and we convert them into liquids. So the extraction process will produce these biofluids. Sometimes we can collect biofluids directly. Those could include things like urine or blood or saliva. What we then do is take those biofluids or extracts and we put them into analytical instruments. Those could be mass spectrometers, NMR spectrometers, liquid chromatography systems, gas chromatography systems. And we use fairly conventional analytical chemistry tools. They're expensive, but they're pretty standard. Where the real trick for metabolomics has come in has been in the last step. And this is the idea of measuring not pure compounds, but mixtures of hundreds to thousands of compounds. And to do that, we have to use pretty complicated data analysis tools. And so it's the data analysis that's developed over the last 10 or 15 years that has given rise to metabolomics. And this is something that, in fact, here in Alberta, we're really good at. So that's the simple pathway or flow chart for metabolomics. Start with samples, run them through big heavy equipment, and then do lots of data analysis to identify chemicals or metabolites in these mixtures. And that could be as few as a dozen to as many as 10,000 different molecules are read out, identified, and quantified. Now, when we do these analyses, we're using different tools. I mentioned NMR spectroscopy, gas chromatography, mass spectrometry, that's GCMS, liquid chromatography, LC, and tandem mass spectrometry. So there's about a half dozen to a dozen different types of instruments that we can use in metabolomics experiments. On the y-axis, I'm showing sort of the log scale of the number of compounds that can be identified with different techniques. So NMR, you can maybe identify 100 compounds. By GCMS, you can identify maybe 200 compounds. By LCMS, it's possible to identify between 1,000 and up to 10,000 molecules. Now, in many cases, as we identify these molecules, we're not really sure, especially with the LCMS, what they are. So they are more like features, whereas with NMR, almost all the molecules we can measure, we know what they are. We've identified them. Now, there's also a difference in sensitivity. NMR is not very sensitive, whereas LCMS is very sensitive. We're able to get down to nanomolar or even picomolar levels of molecules or compounds. NMR is usually good for 5 to 10 micromolar, and GCMS is sort of in between. We have a real challenge still in NMR to be able to identify all the things we can see with these instruments. They're more sensitive than what we realized, and it still means that many of the things we see by LCMS, we don't have names for yet. We're still trying to identify them. That makes it both interesting, but sometimes frustrating. Now, over the last 2 decades, metabolomics has grown exponentially. This is just a graph of the number of publications that have appeared in the literature with the words metabolomics or metabolome. So last year, there were more than 9,000. We're on track to have probably more than 11,000 papers published. Not only are the number of publications growing exponentially in metabolomics, so too is the dollars flowing into it, funding efforts from many organizations and countries. The numbers and types of companies doing metabolomics is growing. So too is the number of service centers and labs growing to doing metabolomics. And that's all because people are realizing the fundamental utility of metabolites. On the other hand, with genomics and proteomics, the growth is not so fast. And part of it's because those are somewhat more mature fields, but it's also because metabolomics is really good at being translated into things like biomarkers or into tests that can be used in labs or for environmental work or for food analysis. Now, the reason for the exponential growth, as I said, has partly to do with the fact that it's a newer technology, but there's also the general realization now that the metabolome tells us more than the genome. It's also the fact that metabolomics is generally more diverse in its applications. It's not restricted just to living things. We can also analyze dead things. We can analyze inert things like water and air and soil. It's also true that the metabolomics is getting faster. Unlike most other omics, it's highly quantitative, and more it's becoming cheaper and cheaper. There's a real push to making metabolomics very automated, which means that it's even faster and even cheaper. The discoveries that are happening in metabolomics, and there are many, are starting to be translated not only into dollars and patents, but they're also being translated into products. The other thing that's making metabolomics really appealing, even though it's uses expensive equipment, are that there are service labs appearing around the world that can do metabolomics really well, really fast, and really cheap. And one of those examples is the Metabolomics Innovation Center, and I am the co-director of TMIC, or the Metabolomics Innovation Center. So this is a shot of one of the nodes for TMIC on the left side, and the CSIS building at the University of Alberta, with our website, and then our standard pitch about what sort of services the Metabolomics Innovation Center provides. It's mandated to help people, companies, academic researchers, non-government organizations in Canada and actually around the world to provide metabolomic services, to do technology development and scientific research, and also to provide most of the world with the software and databases that everyone needs to do metabolomics. So while most of our dollars and effort and staff are devoted to service and science, what we're mostly famous for is the software and data resources, because without the Metabolomics Innovation Center, most metabolomics research would stop. So this is something that many wouldn't realize, but really Alberta is the hub and the engine that keeps the world of metabolomics going. We have been funded since 2011 by Genome Canada, and it grew from a demand that was starting to appear even 10 years ago for metabolomics services by genomics groups. We were tasked to develop cutting-edge services and advanced technology. Initially, there were just 3 nodes in metabolomics, one in the University of Alberta, well, actually 2 at the University of Alberta and 1 in the University of Victoria. It has since grown. There are now 8 nodes across Canada, and it's now funded not only by Genome Canada, but also by the Canada Foundation for Innovation, making it now a national laboratory. There are a number of labs, and this just illustrates the map with the little circles, green and yellow and red, indicating the location of the different nodes. So we have 1 node in Victoria with David Goodlett, a new node in Calgary with Ian Lewis, 5 nodes at the University of Alberta. So this makes the U of A sort of the hub for that. I have a node, James Haranack, Liang Li, Michael Overdwin, and Jen Reed, who's also here, is heading up the TMIC core node. We have a node in McMaster University and another node at McGill University. So we're in most of the provinces, and we serve not only Canada, but a large portion of the world. There's a lot of equipment. Metabolomics is not cheap. It uses big, heavy tools like NMR spectrometers, mass spectrometers for measuring volatiles, for measuring ions and metals, for measuring organic molecules. For many of these, you might just think of this as a whole bunch of abbreviations, but these are pieces of equipment for doing analytical chemistry and for measuring chemicals, metabolites, down to picomolar concentrations for very, very diverse samples, covering, as I say, from blood and urine to soil and water to plants, fruits, and a whole variety of other things from drugs to cosmetics. Right now, TMIC has more than 35 staff. There's more than $37 million of equipment. To run TMIC, it costs us about $6 million a year. I'd mentioned that TMIC is sort of the engine that keeps the metabolomics world going. We provide almost all of the data and software that everyone needs to do metabolomics. TMIC also has a lot of SOP-driven workflows. It's ISO certified. It offers a whole range of services for targeted and untargeted. It does data analysis and interpretation, and it has among the most diverse tool set for metabolomics. Almost 500 publications over the last 10 years, hundreds of thousands of samples, more than 30 million page views on the web servers and databases each year. And TMIC has also created almost a dozen different companies, either through spin-offs or supporting them in a long-term effort. This is sort of a comparison of the different metabolomics resources or centers around the world. There's some in places like Australia and the US and Holland and the UK. There are even smaller centers in Calgary and Toronto. The TMIC is unique not only for what it offers, but also the diversity of what it offers. And in many respects, it is considered the world's top metabolomics resource, both in terms of its offerings from service side, but also from its capabilities. It's able to analyze up to 10,000 metabolites and able to quantify more than 2,500 of those. But also in terms of its software and databases. As I said, these are the tools that everyone else in the world depends on. I have a short video just about TMIC that explains things perhaps in a little more colorful way than what I've been doing through slides. So we'll start running it and hopefully you can hear it.
The Metabolomics Innovation Centre, or TMIC, is Canada's national metabolomics laboratory. We are a leading-edge, geographically distributed, fully integrated facility. We have been operational since 2011 and offer a number of valuable services and products to researchers in the life and environmental sciences. Our offerings include sample analysis, bioinformatics platforms and data resources, and the sale of analysis kits to enable scientists to conduct their own assays. With the capability to identify and quantify up to 3,500 chemicals, TMIC offers custom-tailored metabolomics services that are many times faster and more quantitative and wide-ranging than many other facilities. We provide fee-for-service metabolic profiling that includes comprehensive metabolite identification, quantification, and analysis for the medical, agricultural, food processing, and environmental testing markets. We are widely known for the data and data analysis resources that we freely provide to the global metabolomics community. Designed, assembled, housed, and maintained entirely by TMIC, these resources include both databases and software tools that receive millions of web hits per year, and include the Human Metabolome Database, or HMDB, Drug Bank, FoodDB, Metaboloanalyst, and many others. TMIC also offers custom bioinformatics support on a fee-for-service basis. TMIC scientists continue to convert many of the most popular metabolomics assays into discrete kits that permit the rapid, inexpensive measurement of popular or high-value metabolites. These kits include all of the reagents, software, and protein metabolites in tissues or biofluids. We invite you to visit our website and contact us to explore how we can meet your metabolomics needs.
Okay, so apologies for some of the choppy audio there, but hopefully that gives you a little bit of a picture of what's inside TMIC, some of the tools and equipment. Some of the other things that are not so visible, as I say, are these databases and resources, which are used by literally millions of people a year. These are databases that are mentioned, like the Drug Bank or Human Metabolome Database. Some of them are pathway databases. Some of them are more specialized for food or plants or pathways or contaminants, microbes and toxic exposures. These are kind of like encyclopedias or Wikipedias, if you want, for metabolomics, and they're very rich in terms of the data they have. And many companies make use of them. And in fact, companies have been spun off to resell some of the data and data resources that are in these. Also mentioned in that video were the kits, and these are just some examples. There are kits that are made for NMR, kits for GCMS, and kits for LCMS. And there are different varieties for different platforms. And these are being used around the world as well to facilitate metabolomics. Just like if you're familiar with molecular biology, a lot of molecular biology is now in a kit form. And so by putting metabolomics in a kit, it makes things more consistent, more reproducible, and in many cases, a lot easier. We've also been working on the development of handheld metabolomic devices. This is an illustration of an impedance system that's being developed. So as I said, many of the tools we use in metabolomics are very big and expensive. They're about the size of a large refrigerator. It would be nice if we could do metabolomics on your cell phone, and we're getting closer to that. The systems we have are probably not, they're about the size of a toaster, but they would be very inexpensive and offer more of a capability or possibility of doing metabolomics very, very rapidly and inexpensively in almost any office, on a job site, or even at your home. So I spent a fair bit of time sort of introducing people to metabolomics. Some of you may find this, kind of old hat. Others, this may be the first time you're hearing about it. What I do want to do is maybe illustrate some applications of metabolomics, just so that people can try and link with what is it to what it can do. And I think most of you would be more interested in what it can do for you. So there are lots of applications, both in biomedicine, in environmental monitoring, but also in agriculture and food. So in biomedical applications, there's a number of areas where we've been using metabolomics, helping to characterize the microbiome, to study organ transplant and immunometabolomics. Metabolomics is also used in imaging, what we call chemical imaging. It's used in a lot of diagnostics and precision medicine and patient stratification. There are good examples of how metabolomics has been used to discover drugs and drug leads. In the area of health and wellness monitoring, metabolomics is being used. One area which most people don't realize is that anyone under the age of 25 has probably had a metabolomics test. That's part of newborn screening. And this is where they look at blood spots collected from a heel prick of newborns to identify any metabolic disorders or genetic disorders. Metabolomics is also being used by many drug companies in clinical trial monitoring. So lots of applications from discovery to development to testing. These are some examples of how we've used within TMIC metabolomics to predict diseases, where we might take either a blood sample or a urine sample. We've done this for expectant mothers to identify or predict those who will develop preeclampsia. We've looked at blood samples from expectant mothers to identify fetuses with congenital heart defects or trisomy 21 or 18. We can also use urine from individuals to predict those who are at early stages of cancer, whether they'll develop cancer cachexia or muscle wasting or not. So these are examples where we can look at combinations of metabolites in blood or urine and make remarkably accurate predictions. I'm using these curves, that are called area under the curve, for what will happen or could happen with an individual. Anytime you get an area under the curve of more than about 70%, you're kind of hitting a home run. And in this case, this is what we're seeing of 98, 99 in some cases for metabolomics. You can also use metabolomics not only to predict disease, but you can also diagnose disease. So these are applications, again, that we've used in TMIC and metabolite profiles of blood or urine to detect organ transplant rejection, to distinguish between systolic heart failure and diastolic heart failure, to identify and diagnose chronic fatigue syndrome, or to even pick up polyps from a urine sample. And I'll explain the polyp one a bit more later on and show you how this has been translated into a real-life medical application. We've also used metabolomics to help identify and distinguish people who have COVID and those who don't, as well as people who are sick but are COVID negative and are COVID positive. And in fact, a single metabolite can distinguish, taken from the blood, between someone who's COVID negative and COVID positive, even though they're sick. And this suggests there might be even more powerful tools using metabolomics than the traditional antibody assays, which have a pretty high false positive rate and a high false negative rate. So metabolomics is not just for things in the past, it's things that are of urgency now, where it can be used to distinguish, as I say, COVID positive and COVID negative patients. Agriculture is a big business in Alberta, and because of that, TMIC has been involved in many areas of agri-food applications. It's been using metabolomics in food analysis, herbicide, pesticide testing, in veterinary analysis for cattle and sheep to do disease diagnosis, to assess residual feed intake. You can also do chemical imaging from plant and animal samples. Soil analysis, soil microbe analysis. It's been also used to help formulate foods, areas of precision nutrition, again in wellness, diet monitoring, and there's also work now in doing wine and beer analysis. So one area where metabolomics and TMIC has been involved is the characterization of cannabis, because it's now a legal crop. There's a lot of interest in what is in cannabis, what's good, what's bad. And through efforts with TMIC, they've developed a number of assays to look at the terpenoids in cannabis, the cannabinoids, the polyphenols, the trace metals. It's also compiled a database of everything that we know about cannabis, including the 6,000 odd chemicals there, their concentrations, the reference spectra, and lots of literature data about both the good and the bad with cannabis. Obviously, in addition to plant analysis in agri-food, TMIC also does analysis of livestock. It's worked a lot in the area of cattle, both beef and dairy, and it's helped identify a number of markers for what's called residual feed intake, or essentially the efficiency with which cattle can gain weight. And markers and profiles have been developed that essentially are predictive of residual feed take, not only for cattle, but also for sheep and other animals. TMIC has also looked at other crop plants like brassica, mustard and canola, and to help it distinguish among different strains and varieties that are salt tolerant and are not salt tolerant, and what are the characteristic features of metabolites that allow or protect certain plants from salt tolerance, as well as what protect them from drought. Similar applications have been done for other types of crop plants and also even for conifers and trees. So metabolomics kind of knows no limits in terms of the types of samples, the types of plants or animals that we look at. Metabolomics also is being used in environmental testing and environmental monitoring. I'd mentioned the applications in forestry work, lots of work in water quality and contamination, lots of work in monitoring wildlife and wildlife conservation, particularly with chronic wasting disease. People have also used metabolomics to analyze oil and oil sands products, exposures, both of people, workers, plants, and animals, herbicide testing, metal testing, toxic metals, looking at the microbiome of the soil, bacterial fingerprinting, looking at volatiles, things that cause things to smell, both unpleasant and pleasant. Those are all examples of applications. TMIC has developed very sophisticated methods for pesticide residue analysis as well as herbicide residue. Tools and the techniques are very broad, comprehensive, and quantitative. It's also been used to look at water and the surfactants and actually the surprising number of illegal drugs that end up in our rivers. And it's identified quite a large number of compounds that you probably don't want to know you are drinking. But this is the power of metabolomics and the things that you can see, especially the ones that you may not want to see. We've looked at not only the river itself, but more recently looking at runoff and looking at some of the volatiles. And those of you who live in Edmonton or even Calgary are aware of sometimes the strong smells that can come during the spring runoff as things, particularly from deposits from asphalt, fossil fuels, and degraded rubber end up in our rivers. We are able to detect these and identify them using, in this case, GCMS, which measures many of the volatiles or the smelly compounds. So again, I wanted to highlight the applications of metabolomics, and over the next few sessions, I'll dive into that much more and talk about some of the specific applications for metabolomics in both medicine and in the environment, as well as in agri-food. I also wanted to briefly talk about translating metabolomic discoveries into practice. I'd mentioned before that TMIC's been involved in both creating and supporting a number of companies in Alberta, in BC, in Ontario, and elsewhere, Quebec. These are some of the companies that have spun out. Some of them are, staff of 4 or 5. Others, like Omics, have a staff of about 50. Some of the companies are valued at a few $100,000, others are valued at tens of millions of dollars. But these are all companies that were helped or launched either through the technologies in TMIC or enabled by the technologies in TMIC. In many cases, TMIC has helped with identifying or validating biomarkers for either medical applications or environmental applications or agricultural applications. Other cases, TMIC has helped design and build custom assays for these companies. In other cases, it does large-scale services for the companies. In some cases, it's developed software or databases for these companies. In other cases, it's helping them build their devices, or working on formulating food products, or creating kits for them, or synthesizing chemicals, or offering training and support, or even in many cases, we help companies write their grants and their business plans. So we've played a role for many years in helping make sure that there's a flourishing business, both in metabolomics, but in other areas in Canada. And we're certainly here to help people in that realm. One example, which I think is instructive in terms of what metabolomics can do and what TMIC can do for companies, is this example of the translation of discovery that happened a few years ago concerning colon cancer and colon polyps. So polyps are the early stage 0 or stage 1 form of colon cancer, and a polyp in your colon can grow, and if it grows too large, it becomes cancerous. Colon cancer is one of the most lethal and most common cancers in Canada. It's the second leading cause of death for men and third leading cause of death for women. However, if you catch colon cancer at stage 1 and 2, sort of the polyp or late polyp stage, cure rates are essentially 100%. So it's probably one of the most treatable cancers there are. The problem is that we don't really have a cheap or an effective way to screen for colon polyps. Currently, the only way we can do that is through colonoscopies. And if anyone's ever had one, they know how unpleasant they are. They also cost a lot, about $1,000 each. You can also do things like fecal occult blood testing, but those are essentially for finding cancer at the late stages, which is usually too late. There's things like fecal immune tests or Cologuard that looks for DNA. Those are also typically finding colon cancer, but they require fecal samples. And there's a very low uptake rate of people sending in fecal samples or collecting fecal samples. And so gastroenterologists have been looking for a method of maybe, if we could just spit into a tube and that would tell us whether that would work. And so Richard Fedorik, a former dean of the medical school at the University of Alberta, came up with the idea of maybe we could find a marker for colon cancer and polyps in urine. And in 2014, his team, working with Genomics, used NMR to find us about 6 or 7 chemical markers which were sufficient to identify polyps in urine. So urine test is imminently more feasible and preferred by most people. And a company called Metabolomics Technologies Inc. was started, which was intending to market this, this polyp marker, and they called it Polyp DX. Now the problem was that NMR is not used in the clinic. And so that became very evident about 2 years into MTI's lifetime. And so they approached us in 2017, the Metabolomics Innovation Center, and asked if we could help them develop and modify their NMR assay into a mass spectrometry assay, and one that would be both faster and would use fewer metabolites. And in fact, we were able to do that. We were able to simplify the assay from 6 or 7 down to 3. And we were able to show that the performance was just as good as ever, and that you could do this in urine, and you could do this very, very rapidly. So that test that TMIC helped develop was then picked up by Metabolomics Technologies Inc., and then they marketed it as a laboratory developed test, or an LET, to a number of labs in the US, Atlantic Diagnostic Labs, Lab Express, in the East and West Coast of the US. And so since 2018, the Polyp DX test has been available for people primarily in the US. And over the last number of years, they've been working to try and get the test accepted both in Alberta and in Canada. One of the requirements to get the test accepted was that it had not to only detect polyps, but it also needed to be able to detect later stage cancer. So TMIC has worked with MTI to develop new markers for colon cancer detection in urine. Also working to improve the urinary biomarkers for polyp detection to increase the sensitivity and specificity. In addition, TMIC has been working with Metabolomics Tech Inc. To help convert existing mass spec assays to handheld devices, making these tests something that you could potentially do in your bathroom or at home or in the doctor's office very inexpensively, but also to use these tests in developing countries where access to colonoscopy equipment is almost impossible. We've also been working with MTI to develop very low-cost methods to store, preserve, and transport urine so that urine samples could be collected and shipped easily and inexpensively to testing centers. So these are examples of how both metabolomics is used to discover new markers, which can help in the areas of cancer and precision diagnosis, but also how TMIC works with existing companies to help solve the problems that they have in terms of improving their markers, developing their tests, converting them into new devices or instruments, or helping with the issues of logistics of sending samples around. So I think to wrap up, I mean, this is partly a pitch for Metabolomics, but it's also a pitch for TMIC, and to sort of ask, you know, are there things that TMIC can do for you? Certainly, TMIC, through its many nodes and its expertise, can offer lots of advice on experimental design for metabolomics, can do data analysis and interpretation. It has a very broad array of analytical chemistry services, but it also has excellent programming, software programming teams. It does lots of database development for groups. And it also will do complementary or free pilot studies to help groups, some cases, do exploratory research, or even to help launch some of their business ideas. So if you want to have more information about TMIC and want to talk to people who know about TMIC, you can get a hold of Jennifer Reed. She's TMIC's operations manager. She's also wearing the hat as the business manager. And she's been with TMIC for many, many years. You can also contact me. My email was also at the beginning of this, or you can visit the Metabolomics Center website. So with that, I just want to thank the node leaders at TMIC, Liang Li, who's a co-director, Christoph Borscher, who's also co-director, Phil Britz-McKibben, Michael Overdwin, James Harnack, Dave Goodlett, Ian Lewis, and our funders. And also thank you for listening.
Thank you so much, Dr. Wishart. We do have a couple of questions that have come in. You mentioned in your presentation that up to 10,000 molecules make up the metabolome. Where's the field in terms of identifying and quantifying metabolites at this scale?
So right now we know that the human metabolome probably has more than 120,000 metabolites. Within TMICs, there are assays that we can measure about 10,000. That is, we can putatively and positively identify about 10,000 molecules. And then about 2,500 to 3,500 can be absolutely identified and absolutely quantified. So those are sort of the numbers that we're working with. So we're not able to detect everything, but we are able to detect a lot. And in terms of the number of compounds that we're able to analyze compared to just even a few years ago is probably increased to a factor of 5 or more. So it's growing quickly, the number of compounds we can detect and quantify.
Wonderful. Another question that came in was, how does TMIC support companies in Alberta or anywhere in Canada if they wanted to explore whether metabolomics could address a particular challenge they are facing?
So, I mean, there's several routes. Obviously, as I say, the first thing to do is contact us and to talk with us. We often, whether it's for routine things or exceptional things, we usually have a discussion to sort of just find out what the specific needs are. We do obviously fee for service, but we also do technology development, sometimes through contracts. Some cases we deal with very small companies that are just, you know, sort of a mom and pop operation, and they just want to try something out. And so we've done pilot scale studies with them, a few samples sort of to show this is what you can get, this is how you can analyze it. If you think this is worthwhile, maybe you want to scale up. So, I mean, we've worked, as I say, with small mom and pop companies. We've also worked with, you know, super-sized companies like IBM and Roche and others. So it's a full range, and we've done small-scale pilot studies to large-scale service studies to help with, in some cases, moving the idea into something that's more robust, something that they could actually start marketing a product with. Some cases we've used the pilot information to help them get grants from Western Economic Diversification or other funding agencies. But our mandate really is to help small companies with their ideas. And we've done that a lot and would hope to do that in the future.
Excellent. We do have one question that came in. It says here, has AHS or any other health authority in Canada adopted metabolomic tests for cancer screening or prenatal screening tests developed by TMIC?
So the furthest along right now is the tests for polyps. They're going through, I guess, sort of later stages with AHS approval. In Quebec, tests developed by TMIC have been used as part of blood tests and screening by, I guess it's Optic Lab or whatever it is based in Montreal. So some of these discoveries, yes, are being used, or the tests that have been developed are being used in health services, but they're just at the beginning stage. So hopefully a few more will show up in the next couple of years.
Right. Are there any training opportunities within the TMIC?
We do. We have something called a research hotel, and before COVID, we would be training, or used to train somewhere between 10 to 20 people on site each year. We also have online courses, which we still offer and have been offering. There's a metabolomics course coming up, I believe, in June. And through those, we've trained somewhere around 300 people over the last 10 years. So we do quite a bit of training, whether it's on site or through courses. Sometimes it's how to use instruments. Other cases it's how to use the software and databases. But yes, we do have lots of training, teaching, outreach programs.
Excellent. Large amounts of data would be generated through metabolomics. How do non-experts who would like to use metabolomics deal with these large data sets?
So again, we've been working really hard creating data resources and software that are really, really easy to use. And we put a lot of this on the web. And we do lots of testing so that they're easy to use. So an example is a tool called MetaboAnalyst, and it's used by thousands of people every day. And this is how a lot of people do their data analysis. We also have a team of bioinformaticians who work with different clients from TMIC, and we will process their data, some cases, in a custom way or a more detailed way. And often the data analysis is included as part of the service. Other cases, people just come to us with their data and say, can you make sense of this? If it's simple, we'll just give them a quick answer. If it's really complicated, we'll usually try and sign a contract because it'll take a fair bit of our time and resources. But we understand that this data is complicated, and certainly we're trying to make it easier to analyze, easier to look at, easier to visualize, and we've created lots of resources for that.
Right. We had a question that came in from a gentleman, and he asked here, can we apply for internship in TMIC?
So currently the best way to work with TMIC is through the research hotel. If you want to spend, we've had people spend as short as 1 week and others up to 1 year in TMIC doing things. Some cases they're partially funded by their organization or their supervisor or their company. Some people have self-funded. We do hire people, obviously. We're an organization, and we post ads and positions if people are wanting to actually work in the area. But if you're wanting to train or learn about it, the best thing to do is, acquire a few skills through either, college or university. Once you have those skills, so that you kind of know your way around a lab, then you can come work at TMIC through the research hotel. And hopefully in the summer, late summer, when COVID is mostly over, we'll open the doors again to the research hotel, and people can work or learn in the TMIC facilities.
Wonderful. Just one last question. Can you provide an example where TMIC worked with a company to address a particular challenge they were facing, and how Metabolomics was able to contribute to the solution?
Yeah, well, I think, I mean, I gave one example with the challenges that Metabolomics Technologies Inc. was facing, but we've worked with other ones. There's a company called Metasci in Toronto. They were wanting to distribute metabolites, chemicals, but they didn't have access to a chemical library. TMIC has one of the world's largest chemical libraries, and so we were able to show them the protocols and processes for assembling chemical libraries, packaging those things, how to send those things out for them. Also showing them how to do reference-based work. We even helped synthesize a bunch of chemicals for them. And so because of all that work, then they were able to sort of establish a center in Toronto where they're now selling metabolite libraries around the world. So it's an example where, you know, they were challenged. We had various solutions. We helped them, you know, start walking and eventually even running to the point where they could work independently. Another example, I guess, is Molecular U, which is based in Vancouver, but it also has branch offices in Edmonton. They were trying to find ways of doing nutrient analysis and food and diet analysis. And they couldn't find a group that could do that. So we developed a bunch of assays for that. And now we're able to do that for them.
Oh, that's fantastic. Well, we're getting close to the end of our time together. So in closing, I'd just like to remind everybody that BioAlberta relies on the support, collaboration, and engagement of our members for us to deliver industry development related initiatives like this webinar, so that we can be an effective voice for Alberta's life sciences sector. And we hope that you enjoyed today's presentation. If you're interested in joining our ranks, we offer corporate and individual memberships, which are detailed on our website at www.bioalberta.com/member area. And on behalf of BioAlberta, I really want to thank Dr. Wishart, co-director of the Metabolomic Innovation Center, and their business development manager, Jennifer Reed, for their time and expertise in bringing you this excellent webinar offering today. And thanks to all of you for joining us. This event has been recorded and it will be available on our website. Should you have any other questions that you'd like to maybe put forward for Dr. Wishart, feel free to, I'll put my email address in the chat here after I finish talking. But feel free to send me an email and I can certainly get those to him and we can get you answers. But this has been just tremendous. Dr. Wishart, I can't wait till we have the next three, and registration is open for the next three events. The next one is going to be on the 12th on precision medicine. And so I hope that everybody who's joined us today can join us then as well.
Thanks so much, Tricia. Thank you, everyone.
Thank you, everybody.